Good day, grade 12 learners. Welcome to today's Life Sciences revision lesson. My name is Sina Sekheche, and this lesson is brought to you by the Gauteng Department of Education in collaboration with Saibono Discovery Center. Right, so for this lesson today, I will not be alone. I will be with my colleague, Shabnam Sukmandan. Right, so let's just get into it. I hope you are ready for your exams that you are writing tomorrow. You have studied. Today you are just putting a covering on everything that you've studied so far. So let us take a look at the questions or at the weightings of the topics for your paper. Right, you have done DNA code of life. So in your paper, that will be amounting to 14%. That will give us 20 marks. So out of that 150 paper that you'll be writing tomorrow, 20 marks will be the DNA code of life. And in meiosis, it will be 16 marks out of 150, which amounts to 11%. And then reproduction in vertebrates, this is a smaller topic. So because we are half yearly, you are only going to be assessed for 4% of this paper, and that amounts to 6 marks. And then human reproduction, this is quite a lot. This is 21 marks. 21%, that will be amounting to 31 marks out of the 150. And in genetics and inheritance, 24%, that will be amounting to 36 marks. And in responding to the environment, that is the human response to the environment, it is 28%, that will amount to 41 marks. So if you take a look at these marks here, you see it helps you where to put your time or where to put more time in studying as you dissect your topics according to the exam guideline. So let us check what are the things that you are going to need for your studying. I know that you have used some of these resources already, but if you haven't, just check if you have all of this. This will help you to study everything that you need for your life sciences paper tomorrow. Right, you must have a DBE study guide. This was sent to you maybe by your teacher, or it's also in the Google Drive for Life Sciences. And you must have the SIP material. The SIP material for the previous years, they have beautiful questions and beautiful summaries that you can use there while you are studying. Previous exam papers, trial examination, the May-June examination, or the February-March examinations. So if you have the previous question papers, you can use those to study per topic. Then the notes and the classwork activities that you've been using during teaching and learning in your classrooms. Your teacher must have given you some notes, or you shall have written some notes on your books or on your workbooks. You use those together with the exam guideline. This is very, very important. The exam guideline, very important, because this is the document that is going to show you now the scope of how much you need to study per topic. And then Mind the Gap. Mind the Gap is a wonderful document that has simplified versions of these topics. It has summarized them into smaller chunks that you can be able to use if you are struggling with a whole lot of information at once. So you can use Mind the Gap for that. They have beautiful explanations of processes that you can see, and also beautiful pictures that you can see. Then last but not least, you can use the internet. You can go to the Department of Education or education.gov. You can also use Pocket School. Pocket School lessons, the ones that we are doing now is a revision lesson. However, you can go to Pocket School and check all the lessons on all the topics that were done in term 1. That will prepare you for your examination tomorrow. Right. And for this lesson, please remember that we are only going to look at some of the more difficult concepts. We are not going to do everything because we don't have much time. So we are going to do only the difficult concepts per topic. So let's just get on with it to save time. Right, remember for this paper that you are sitting for tomorrow, DNA is going to amount to 20 marks. Now, what are the key points that you need to remember on DNA or nucleic acids? First, you must know the history of the discovery of the DNA. You must know the role that these scientists played. Watson and Crick Franklin, and Wilkins. Remember, this question may appear in your match column question, in your multiple choice question, so you need to know the years, you need to know who discovered DNA, who took the X-ray pictures, who won the Nobel Prize. Please, and also check the years. So the history is therefore important. Then, the second thing that you need to know is the structural difference between the DNA and the RNA. In your workbooks, you must have activities where you were dealing with the differences between the DNA and the RNA, where it is found, the nucleotides it has, on all the nitrogenous bases, maybe that differs. So you need to be able to differentiate between DNA and RNA in that sense. Then the differences between DNA, mRNA, and tRNA are important. This now is going to prepare you for when you are doing protein synthesis. Then you know, when do we find DNA, when does it become mRNA, and when do we find tRNA, and what roles do each play in protein synthesis. So the difference of knowing these three is quite important for you. Then you must also be able to differentiate between DNA replication and protein synthesis. These two processes are happening, but however, they are not the same. So you need to be able to say, when I'm talking about DNA replication, is when the DNA is making exact copies of itself, and how that process unfolds. All the steps following the logical flow, you must be able to define this, and you must also be able to define protein synthesis with its two stages, and be able to define using a logical flow of events as how it happens until we have formed that amino acid, that is going to form proteins later. Then these are the two processes that protein synthesis takes place in. You must learn transcription and translation. This is where I'm talking about those logical flow of the steps that you need to know. But don't worry, we'll look at it. We're going to look at some of these things as we go forward. This is just for me to help you to remember what are the key things that were covered during this topic throughout the year. Then DNA profiling is important for application questions. So here you must be able to interpret a DNA profile where you use a ruler. You must have that skill and know the reasons why or where do we use this DNA profiling. Then paternity testing, which involves both parents and the child. You must be able to do that while you are analyzing this DNA profile. Also uses the interpretation of DNA profiling. So all of this you must be able to do. I think by just going through these key aspects, your mind is now coming back to remembering what are the things, or what are the key things that you covered when you were doing this module. Right, let's get into it. So the first thing that most of you are struggling with is protein synthesis. Learners take protein synthesis as if it's something long that they could not understand. You need to break it down. So let's break it down for you tomorrow to remember. Remember, what is protein synthesis? Protein synthesis is a process whereby DNA molecules make copies of itself and decodes to form what later? To form a protein. So how does that happen? It does not just form a protein. There are steps, there are processes that happens, there are many organelles, enzymes that are involved. So let's unpack it. How does protein synthesis take place? First thing that you need to know is it takes place in two stages. So we will say this is our first main stage, which is transcription. And transcription happens where? It's in the nucleus. So your transcription will be here. Right? Excuse my handwriting. I just want to write fast. And then the second stage is going to be translation. Some of you are confusing these stages. How do you remember which one comes first, which one comes later? This is how you can remember them. Look at this term, translation. Lation. You can say it sounds or rhymes with later, meaning this is the stage that comes later. That's how you can remember it. And this one happens where? In the ribosome. In the ribosome, you can say it's just later. The first one is transcription. This is where now the coding of mRNA is going to take place. mRNA is going to form. So let's take a look at how mRNA is forming. The first one, this is very important when you describe the steps. They must follow a logical flow. Please, please, please make sure that your story makes sense as you go on and explain. Right. The first thing that must happen, you see this DNA. This is our DNA. It is a double helix structure. It cannot do anything if it is still double helix and twisted like that. It must do what? The double helix must unwind. What is important here? Please make sure that you say the double helix unwinds, not just DNA. Just say double helix to be technically correct. Then the weak hydrogen bonds, now you are following the flow. The weak hydrogen bonds between the nitrogenous bases do what? And two DNA strands, they unzip and separate, or you can say they break. Remember, for all of this is just 1 mark. For saying the two hydrogen bonds break, it's 1 mark, and then the DNA double strand unzips, it's 1 mark. So unzipping and breaking, it will give you only just 1 mark. Then 1 DNA strand. Now, this is very important for you to differentiate with DNA replication. Remember, the first stage of protein synthesis, it is similar to DNA replication. However, there are differences. And now we are coming across the first difference. What is the first difference? It is that the DNA strand 1. Please take note. Remember, in DNA replication, you say the 2 strands. But here you say 1 strand. So it will be just 1. If you are replicating this DNA molecule above, only 1 strand acts as a template for the construction of mRNA. So meaning it is the only one that is going to be exposed and act as a template for that formation of mRNA. And then how is that going to form? How is the mRNA going to be formed? Remember, when it is a strand acting as a template, it is like this. It has its nitrogenous bases exposed. So what is going to happen? The free RNA nucleotides from the nucleoplasm form mRNA, meaning if it has exposed the nitrogenous bases like that, the free floating here RNA, this is another difference from protein synthesis, because from DNA replication, because in DNA replication you talk about the free floating DNA nucleotides, because you are making copies of what? Of DNA. But here, because we are forming mRNA, you want what? Only the nucleotides that form the structure of mRNA. And you shall have studied the structure of RNA, and you know the nucleotides of RNA together with the nitrogenous bases. Yes. Then this is the second difference. The free RNA nucleotides are going to come from the nucleoplasm to form mRNA. So as it comes and attached to that exposed template, it is copying the code from DNA and forming what? mRNA. And that new mRNA that is formed, this one here, mRNA that is formed, what is going to happen to it? It complements that DNA in the nucleus. Remember why it complements? Because the template has exposed what? The nitrogenous bases, and when the nitrogenous bases are exposed and the RNA nucleotides are coming to complement that DNA strand that was exposed, and when it is complementing like that, it is forming what? A new strand of mRNA, and this new strand is going to do what? Now we are moving to the next stage. What is the next stage? Is translation. Translation, we said it happens later. What happens there? The coded mRNA now leaves the nucleus through the nucleopore. So you must be quite specific. You see here, it moves through the nucleopore. You must be able to also label this diagram to say these are your nuclear membrane and this is a nucleopore and this was your nucleoplasm. Then the mRNA leaves through the nucleopore. Then what is it going to do? It is going to attach to the ribosome. You see this arrow here of this red. It is going to move and attach itself on the ribosome. Let's write it. RNA. Remember when you're writing RNA, mRNA or type of RNA, the first letter must be a small letter and then the rest must be capital letter. Even with the type of DNA, remember that is the correct way to write it, and it must be visible when you are writing, especially for question number 1.2 for your terminology. Right, once it attaches there, look at what happens. tRNA is going to pick up the amino acid and complement the mRNA in the ribosome, meaning this tRNA is going to bring the required anticodon. What you must also remember is that the triplets bases that you find on an mRNA are called codons. I think you all remember that. And then the triplets that are found on a tRNA, or the 3 consecutive nitrogenous bases on a tRNA, are called the anticodons. And then the anticodons are complementary to the codons on mRNA. So let us go back to step number 7. What is going to happen? tRNA is going to bring about the required amino acid by doing what? By complementing the codons on mRNA, meaning it's going to pick up what? It's going to pick up all the nitrogenous bases that will be complementing the triplets on mRNA. And once they complement, they release one amino acid. And then these amino acids are going to bond. You see here, they form a chain. Look here. They are going to bond by peptide bond. Remember, the bond here is peptide bond that joins together amino acids to form what? A large protein. So please remember the flow of events when you are doing protein synthesis. So this is where you are mostly getting the marks wrong. Then we move on to DNA profiling. What is DNA profiling? Remember, we are saying profiling, and then profiling is a process, is a technique. Profile is what you have, is the information that is printed, that is in front of you. This DNA profiling, what is it? It's a technique that is used to identify the sequence of bases. Remember, when you are asked for a definition, go to your glossary that you have compiled at the back of your book, go to your terminology test, and also go to the exam guideline. Exam guideline is very important when they are asking you about terms, to define the term. You must define it as it is defined in the exam guideline for you to get full marks. So what is DNA profiling? It is a technique that is used to identify sequence of bases. Now, the nucleotides are separated from each other in that they are found in the strand of DNA. Now, the nucleotides appear as dark bands. I know you are familiar with this. What you're seeing here on the right, you see this is a sample, a DNA profile. It has all the information that was collected when they were doing what? DNA profiling. Now, when you see these dark bands, it tells you that these are the nucleotides. They are going to appear as dark bands. And then DNA profiling is different from fingerprints. So because here we are dealing with DNA and nucleotides, and it has different types of nucleotides. So what do you do for these types of questions? Like we said in our key points, is that you need to be able to analyze a DNA profile like this, be able to tell us who is the suspect, solve crime, or tell us what are the uses of this DNA profiling. And the key thing here that most of you are making a mistake is to just use your eyes and look through this. Remember, it is important for you to have a ruler and use your ruler to match or to compare the dark bands of the nucleotides, so that you know exactly this is your suspect in case this was a crime scene. So you must be able to do that using that. Another thing is you need to study mutation. Remember, we spoke about mutation during the year. You learned about mutation. But there's a question that always comes when it speaks about mutation happening in DNA or in mRNA or in whatever. So let us take a look at this example just to bring you closer to that. Now, there's a scenario that we have here. This scenario says that the table below, or this one alongside, shows some mRNA codons and the corresponding amino acid. Let's go. These are the mRNA codons and their corresponding amino acid. Remember, the amino acids are the building blocks for the protein, and you don't need to study the amino acids. The amino acids, if you are asked a question on amino acids, they will be given to you in a form of a table. However, the table needs to be analyzed. That is my table being given, or are my amino acids names given in the form of mRNA codons, DNA based triplets, or tRNA anticodons, so that you know whether you need to decode back or code forward for you to get the specific name. That is the key thing that you need to look at your table to say, am I given the names of the amino acids in terms of mRNA or in terms of tRNA or DNA? Right. Then it says a section of mRNA has the following base sequence and is read from left to right. So this is the sequence. You have GAU, CUC, GAC, AGC, AUG, and ACC. So that is the sequence on mRNA, we are told. Then what you are dealing with now, a mutation. Remember, this is a typical question that can come. A mutation occurred which resulted in the following base sequence on the mRNA molecule. Now, there was a change in the sequence of the nitrogenous bases. That's how you explain mutation. Go to the exam guideline. Then, here, GAU still the same, CUC still the same, GAC still the same. Now, when we get here, AGU, we see there's a change. Instead of UC here, we now have U. Then we have AUG and ACC. Right. Then describe the mutation that occurred. How are you going to start to attack or to tackle this question? The first thing that you must do, you must compare the original mRNA to the one that has undergone mutation. Meaning you must tell us where the change was, to say on the triplet base or on the sequence of the bases. ACG has now changed. The C was replaced by U. So you must be able to explain what happened. Identify the affected codon or a nucleotide. After you've identified that, describe which nucleotide has been replaced or deleted. So you must be able to tell it from there. So this is the first and the simplest thing to identify and to be able to tell. However, the question, remember, this is the answer. C was replaced by U on the fourth codon. So that was our answer. However, it can go further to the effects that the mutation will have on the resulting protein. So here, you must be able to tell us. What must you be able to tell us? You must use the given table to find out if the new codon formed after the mutation codes for the same or a different amino acid. What do we mean by that? Let's check. What was the first one? The first one was ACG, this one. It coded for serine as an amino acid. So that in that sequence, your first one will be serine. And then it changed to this one. It mutated. Now this was after mutation. AGU. Remember, the C was replaced by U. However, what you find here is it still codes for the same serine. But in other cases, you may have a case where when the mutation occurred, it coded for a different amino acid. Therefore, you are going to write the name of that amino acid. Let's have a look. First, the affected codon AGC in the original RNA codes for amino acid serine. We've done that. We've already identified. Then the second one, the codon that has undergone mutation, this one, where you replaced the C, also codes for what? For serine. So that was what you have identified. So it codes for the same amino acid, which is serine. And be specific. If you have the names according to the table, use the name that is given in the table. The amino acid sequence will not change. It will not change because of what? Therefore, there will be no effect because the same protein is going to be formed. If this was a different amino acid, then it means it was going to form a different protein. Here you are going to write a different protein is going to be formed. You must follow the sequence, right? Then we are done with your nucleic acids. We're moving on to meiosis. Meiosis is going to amount to 16 marks out of your paper. And then remember here, you just need to be able to tell us this is the type of division where a diploid cell undergoes 2-cell division to form what? 4 haploid cells. That is the definition of meiosis. Now, before it takes place, you must be able to tell us that it undergoes what? DNA replication. And then it goes into the first and the second meiotic division. The first one is where you are forming 2 haploid cells. And then the second division, you are forming 4 haploid cells. And then what is the importance? The importance, crossing over introduces genetic variation. This reason you must know for life because it interlinks or it it goes with other topics that you are going to study later. Then it also ensures that sex cells or gametes have haploid number of chromosomes because we want to keep it at a haploid number so that when fertilization takes place, there is diploid. Then there's also terminology. Here is a terminology that most of you confuse when you are dealing with these topics. So the first topic term you must know: homologous chromosomes. What do they mean when they talk about that? Bivalent, chromosome, chromatids, centromere, centrosome. Here, this is where you find a challenge. Please make sure that you know. Crossing over, chiasma, a karyotype, nondisjunction, karyokinesis, and cytokinesis. Please make sure that you know this terminology because it might appear on your paper tomorrow, any of these terms on your paper tomorrow on question 1.2. That is your terminology question. Then meiosis takes place in phases. You have different phases. There are different ways for you to remember these stages in sequence, how it happens. You must use the mnemonics. I know some of you have nice ways to remember this PMAT. That mnemonic tells you that prophase comes first before metaphase, then followed by anaphase, then later by telophase. However, because you are dealing with meiosis, remember to write the numbers. Whether it is stage 1, prophase 1 or prophase 2, metaphase 1 or metaphase 2, anaphase 1, anaphase 2, telophase 1, telophase 2, depending on the events of what you are seeing in the cell. When you look at the cell here where we start, remember the preparation phase interphase happened, and then here now we are doing what? We are starting with the process, which is prophase 1. There is a lot of things that are happening at prophase 1. When you look at the cell, you must be able to identify few things that are happening to confirm to you that this is prophase. So you must know the phases by knowing the events of what is happening by just looking at the chromosome. So you can say now the chromosomes are starting to appear as homologous chromosomes here. The centrosomes are forming, nuclear membrane is disappearing. All those events that are happening at prophase 1. And then you come to metaphase. How are you going to differentiate between metaphase 1 and metaphase 2? Here you know that at metaphase 1, meta means middle. That's how you're going to remember this one. What do you expect to see there? In metaphase 1, you're still talking about homologous chromosomes. So you expect to see the chromosomes in homologous pairs lined up at the equator. That is going to give you that it is the metaphase, and then they are attached to the spindle fibers. Whereas in metaphase 2, here, look what is happening. The chromosomes are arranged singly at the equator. So already you know what is happening during this stage or this phase, metaphase, in the first stage of meiosis and in metaphase 2. Then anaphase, you can just say apart. Here you expect to see things being pulled apart. So in anaphase 1, what do you see there? You see that the chromosomes are pulled apart to the opposite poles at the spindle fibers shortened. Remember, they are attached to the centromere. So as they shorten, they pull the chromosomes to the opposite poles. And then here at anaphase 2, what is the difference? Instead of the entire chromosome being pulled to the opposite pole, what is happening? Now the chromatids are pulled to the opposite poles, and the same, they are attached to the spindle fiber. Then at telophase 1, you find 2 cells are formed. Remember, you must be able to say that the cell wall is going to do what? A cleavage is going to form if you are talking about a animal cell, and then in plant cell you just say they form what? A cell plate. And then further, when you go to meiosis 2, remember this is 1 and this is 2. So when you go to 2, the previous cells that you had are the ones that are going to further undergo this division until you find that at telophase 2 you end up with what? Okay, here I wanted to say 2 cells. Then here you end up with 4 cells or sex cells, which we call gametes. So that is what you need to remember. Please go through the processes fully. And another thing that you need to remember here is nondisjunction. What is nondisjunction? Spindle fibers fail to separate during meiosis. Sometimes, remember, all these processes are controlled, but sometimes things happen. Now, when the spindle fibers fail to separate, what's going to happen? This may lead to members of one pair of homologous chromosomes fail to become separated during which stage? Anaphase 1 or anaphase 2. Remember, anaphase is where they are supposed to separate. In anaphase 1, the homologous chromosomes are supposed to be pulled or separate to the opposite poles. In anaphase 2, the chromatids are supposed to be pulled to the opposite poles. Now, if that happens, if there is non-disjunction of chromosome pair 21 in humans, remember, you must be specific here. Human beings have different the genetic disorders and also some other animals. So here at chromosome pair 21, if there is that failure to separate of chromosome and you end up with an extra copy at chromosome 21, that we call it what? Trisomy 21, and it may lead to a condition called Down syndrome. Then it will have what? 2 gametes will receive an extra copy of the affected chromosome, and then 2 gametes will lack that chromosome. So when you're looking at a diagram that shows non-disjunction, the first thing that you must look at is one gamete is going to have an extra copy, and then another gamete is going to have less, because where it was supposed to separate to make it equal on both gametes, it did not. Then the other one is left with less, -1. Now, when you are explaining Down syndrome, please refrain from talking about a chromosome that is less. To say there will be less, talk about an extra copy, because we want to see trisomy 21. When you are talking about a less number of chromosomes and you are explaining Down syndrome, it is no longer Down syndrome. So we must see that extra copy that makes 3 chromosomes at chromosome number 21. We know that most of you like to answer in negative form. So that negative form might give you negative results. So please answer in a positive form. Right. Then the next topic that we are going to do is human reproduction. This is quite a big topic. It amounts to 41 marks of your paper tomorrow. However, there's a nicer and simpler way that will only take you a few minutes to revise the entire topic. Remember, you've studied the male, the diagram of the female and the male. That is what you must know. You must also know the diagram of the ovum, all the layers, the jelly layers and all that. And also be able to draw the diagram of the sperm cell. And for the male and the female reproductive system, look at the diagrams, be able to know the functions of each. And then puberty, you are going to also to be expected to know this for your test tomorrow. And then events of the menstrual cycle. And menstrual cycle is divided into two: what is happening in the ovary, and also what is happening in the uterus, which is regarded as the uterine cycle. So you must be able to know what is happening in the ovaries, what is happening in the uterus, and know the roles of the hormones that play a role in menstrual cycle. Those are your FSH, LH, estrogen, and progesterone. Then the development of the fetus. Remember the development of the fetus, where you must know the placenta, the villi, and all of that. You must be able to know that. Now let us take a look at this beautiful, simple way to remember the roles of the hormones during menstrual cycle. You can use this acronym here, FOLP. FOLP will help you to remember the sequence of the hormones as they come. So the first hormone is your F from your FOLP. What does F stand for? Your F is going to stand for FSH, follicle stimulating hormone. So let us trace what is happening in our graph to explain the role of FSH during the menstrual cycle. So we go to our graph and we check. When you check the graph, you start from the bottom, you check, you have from 0 days to 28 days here. But from 0 days to about 7 days here, we can say 7. You can see what is happening. There are menses, meaning the person is bleeding from the menstrual menses. Yes. And then what is going to happen? Look at what is happening in the ovaries. There are follicles already there. Then you look at the body temperature. Sometimes it's not there, not important. Then the pituitary hormones, very important because there are hormones that are secreted by the pituitary, and then ovarian hormones, and then there are hormones that are secreted by the ovaries. Now let's check after day 7 what happens. The first one, remember the first hormone is FSH. We go to the graph and look what organ secretes this hormone. It is secreted by the pituitary gland. So you go to the pituitary hormones and you check the level of FSH. Look at the level of FSH now. It is the lighter green one, this one here. I'll put a star on it. So that lighter green one is your FSH. Look at what is happening after day 7. FSH now is being secreted by the pituitary gland and the level is starting to increase. As it increases, where is it going to act? It is going to go and act in the ovaries. Now you are talking about the ovarian cycle. To the ovaries and do what? Stimulate the development of these ovaries. Now the ovaries, remember they were there, they were small. Now they must now develop and grow. They are going to grow from primary follicle, secondary follicle to up until the Graafian follicle. So they grow to those stages under the influence of what? Of FSH. And then look at what is happening. As the follicle is growing, look at what is happening in the ovaries themselves. There is a hormone that is secreted, that is going higher. I will draw a circle on it. That hormone is now estrogen. This hormone, which is our second hormone. Now, estrogen. This second hormone is secreted by those developing follicles. And when it is secreted by these developing follicles, look, the level is going high. And where is it going to have an effect? You go and check in the uterine wall. It's going to now thicken the uterine wall and make it more thicker and vascular. The wall is going to be more thicker and vascular, right? So, and after that, you go, the days go. This is now matured to the Graafian follicle, remember. Now, on day 14, this one, all of you know, what happens? LH. Now, this is our LH, our third hormone. LH is secreted. By what? By the pituitary gland. Look at the pituitary hormones. LH on day 14. We'll put a triangle on it. So LH on day 14 is secreted in high amounts. Not that it was not being secreted. It is secreted now in high amounts. And where is it going to have an effect? It is going to have an effect where? Inside that follicle, that Graafian follicle, and cause that Graafian follicle to rupture and release an ovum. And when it ruptures and releases that ovum, that stage, that phase, that process, we call it what? We call it ovulation. And also, this LH is not only going to cause the Graafian follicle to rupture and release, it is also going to form the development of what? Or to stimulate the development of the corpus luteum. Right, let us check. Once this ovum is released, LH was released, then this Graafian follicle that was ruptured, it is going to form now what we call a yellow body, which is the Graafian, which is the corpus luteum. This also happens under the influence of LH. And now this corpus luteum, look at what happens once it is formed. Once the corpus luteum is formed, you check what is happening in the endometrium. The endometrium is getting more vascular, more thicker. You see, why? Because there's a hormone now that is being secreted by the corpus luteum, which is called progesterone. Now, this is the hormone that is going to maintain pregnancy. So the corpus luteum is that yellow body, is going to continue to grow, going to continue to secrete what? Progesterone. However, if pregnancy did not occur, what is going to happen to the corpus luteum? You look. This is the corpus luteum is going to degenerate. When it degenerates, it becomes smaller and disappears. And when it disappears, no more. What is going to happen to what it was secreting? The level of progesterone is going to go down. You see, level of progesterone now is going down. Why is it going down? Because it is no longer being secreted by the corpus luteum, because the corpus luteum is now has now degenerated. And we can say this person is not pregnant because progesterone is not there to maintain pregnancy. Therefore, the person will undergo the menses, meaning that endometrium wall that was being built and thickened in preparation for pregnancy is now going to shed. When it shed, it's going to start the menses again, and the whole process starts again. So that is how you can track the roles of the 4 hormones in the menstrual cycle. So these are some of the questions that we have developed in the worksheet that you have to go practice. So please download this worksheet and go through this worksheet for these questions to prepare you for tomorrow's test. It is quite a wonderful question. I would like for you to go through that question. And now here, you need to know the stages of the development of an early embryo. This here, that after fertilization, there is going to be a zygote formed. That zygote is going to divide by mitosis to form what? 2 cells, 4 cells, 8 cells, until it forms a ball of cells, which is called a morula. And then that ball of cells might have about 18 times 2, 16 cells. And then you have that ball of cells, when it grows again and then it opens a halo here, we call it a blastocyst. So you must know, from a zygote to these numbers of cells by mitosis, to morula, which is a 16 ball of cells, and then to a blastocyst, which is a hollow ball of cells. Now this is now your embryo developing until it attaches to the endometrium. Once it is attached to the endometrium, what is it that you need to know? You need to know the roles of all these layers that are covering. How is this fetus here that is developing protected? And how are the blood vessels supplying the nutrients and the oxygen? Please read about the amniotic, the amnion, the chorion, and remember, you don't confuse this with the choroid of the eye. You're going to learn about the eye now soon with Suki. And then the chorionic villi, also just note, and the placenta, the roles of all these layers in the protection of the fetus. Right, and then this was also another question that you can practice that says, this is a very good activity that is going to help you to understand those layers. Can you please go through this activity because it has those structures? It says, describe the structures that develop after implantation to ensure the protection and the nutrition of the developing embryo. So this one is the one that is going to take you through all those layers that are protecting the developing fetus. Please attempt to answer this question on your own, and then I'm going to leave it there for my colleague Suki to take you through genetics and inheritance, human response to the environment as well. Thank you so much.
Good day, grade 12 learners. Welcome to the second half of the mid-year exam revision program. And we're going to start off with a very important topic of genetics, which takes a lot of marks, 41 marks for this exam, and like about 48 marks for end of the year. So what we're going to look at, learners, is we're going to look at the concepts of genetics, which is your genotypes, phenotypes, what's homozygous and heterozygous, okay? As well as how do you do a monohybrid cross, right? Dealing with just one genetic characteristic, how you actually use the alleles for a gene. Remember, you must know the difference between a gene and an allele. Then we're also going to look at how you do a dihybrid cross with two genetic traits. And we're also going to look at sex-linked. How will you answer a sex-linked disorder, which is not always caused by a recessive allele. It's sometimes caused by a dominant allele. Therefore, you need to read your questions very well. We're going to look at those circles and squares that look very, very scary. But actually, if you know that your squares are males and your circles are females, then you just have to work backwards to look at the genotypes. We don't have much time. We just have 45 minutes, and therefore we would not be able to look at stem cell research. But remember, you need to know this for the exam. What is the definition of a stem cell? And that is any cell that can change into any kind of a tissue or differentiate into any kind of a tissue, then you need to know the importance of genetic engineering, right? Like how that it increases the food life, it gives the food a better taste so that more can be sold. And remember, with genetic engineering, when you're looking at any question that deals with explain, you got to give a statement and a reason. For example, genetically modified food, right? It's expensive and therefore only the rich can afford it. It improves the taste, therefore more can be sold, increasing profits. That's how you do an explanation question. Then we're also going to look at how do you actually test for paternity. Paternity testing means how will you know whether this child belongs to that parent. So we have two ways in which you can test it, either a DNA profile or a blood group. So that's an overview of what we're going to be looking at. Right. So here's your terminology, which often comes out in section A. Remember, chromatin deals with a ball, a network of chromosomes. And a chromosome is just a single strand. Right. We have 46 chromosomes in our body cells, but we have 23 chromosomes in our gametes. We have 23 pairs of chromosomes in our body cells, but we have just single 23 chromosomes in the gametes. That's how you need to know them. What is a gene? It's a trait. It's a genetic trait. Blue eyes. I'm tall. I'm short. I have gray hair. I have white hair. I have short fingers. I have rounded nose. I have a stumpy nose. I have a long nose. I have a round chin. All those are genetic traits, okay? So that is a gene. That is how you describe it in words. But now to represent a gene, we use a form. We use an alphabet, which we call alleles. So alleles are forms of a gene, but in a genetic cross, you represent the genes with alleles using alphabets. It depends. Whatever you see on the outside of a person, let's say if a person is tall, then that will be a dominant allele. So if there's two tall parents, but the children are short, it means in the parents, tall is dominant. But suddenly the child is short. It doesn't mean that the parents did something wrong where the child became short because the mother must have had an affair with the short man. No, it means that both the parents were tall, but one of them or both the parents are carrying the allele for short. It means the mother and the father's parents must have been short. And that's how the allele gets transferred from one generation to the next. Remember, genetics is a study of inheritance. All right, so allele is what? It's a form of a gene, and we use alphabets to represent it. If you can see that gene in the phenotype, the physical appearance, it's dominant, you'll use a capital letter. If that allele is there, but you cannot see it in the phenotype, in the outside appearance, it means it's recessive, and we represent it with a small letter. That's dominant alleles are big letters, recessive alleles are small letters, and they are hidden traits. You don't see it in the physical appearance. Phenotype means what you see on the outside, the physical appearance. We use words to describe it. A tall man marries a short woman. A red apple is crossed with a yellow apple. That's phenotype. Genotype means we must use alphabets to represent it, okay, like a big T or a small t. Homozygous means we use the same alphabets. That means the two capital letters will be homozygous dominant, right? Like that. Two small letters will mean it is homozygous but recessive. Hetero means different. It means one big and one small. It means that this person got a dominant allele from the one parent and a recessive allele from the other parent. Always remember, an individual inherits one allele from each parent. Monohybrid deals with one genetic trait, just one. And dihybrid means two genetic traits. Okay, so that is an overview. You have to know, learners, the format of doing a genetic cross. If you know it, but you put the words in the incorrect place, you can lose 6 simple marks. So this is the layout of a genetic cross. You can put P1, that's the first generation of parents. Then you must describe what they give you in the question. You write phenotype and describe it. Is it a red apple that's crossed with a yellow apple? Is it a man with brown eyes that's crossed with a female with blue eyes? Right? That's the phenotype. You use words. Then the genotype, you'll always get it in the question. They will give you an alphabet. And what you must know, you must look out for the big alphabet. Oh, the capital letter represents a dominant allele. The small letter represents a recessive allele. And then you're going to represent your genotype using alphabets. Then after that comes meiosis. Put it on the next line separately. Then comes your gametes. You cannot have your gametes on the same line as your meiosis. Meiosis comes first to produce the gametes. Okay. So you're going to write your gametes there, and then you do a cross, or you can also do a Punnett square. Okay, learners? You can have 3 columns and 3 rows. In the top left-hand corner, you write gametes. Okay, and you put 1 gamete per block like that. And then you would cross it like this. If that gamete is fertilized by that in that line, big B, this is homozygous dominant. If this gamete is fertilized by that one, it's going to be big B, small b. Always represent your capital letter first. Then you've done the first allele with the second, or first allele with the first, you put it there. First allele with the second allele of that gene, you put it there. Now you've done with this allele and these 2 alleles. Then let's look at this allele. What happens if this allele of this gene is crossed over with that? So in that line, big B, small b. And what happens if this allele was crossed over with that during fertilization, small b, small b? What does this Punnett square or this cross represent? In line, it represents fertilization. You get a mark for that. So these are your offspring's genotype, and that is what fertilization occurring randomly. Then you're going to write down, now you must describe this genotypes. You'll write the F1. These are the offsprings of those parents, F1, and you'll describe it. For example, here now, if big B was black and if small b was blue, it means 1, 2, 3. See, each block represents 25%, or 1, 1, 1, 1, 1. 1 out of 4, 2 out of 4, 3 out of 4. So now, this, if it's a percentage, that means this is 25%, 25%, 25%, 25%, giving you 100%. So how will I describe the genotype of these are my zygotes now, that's formed from my gametes? This is going to be what? 1 homozygous black-eyed parent. This is going to be another black-eyed parent, but it's going to be heterozygous because it's capital B and a small b. That's also going to be a capital B and a small b, also black-eyed, but now heterozygous, right? So it's going to be 1, the genotype will be 1 homozygous black-eyed, and is to 2 heterozygous black-eyed is to 1 homozygous blue-eyed. You see, in genotypes, you represent it as homozygous or heterozygous. Trying to say, is it capital letter, 2 capital letters, or 2 small letters, all right, or 2 different letters. That's why you say that as a genotype. But now, phenotypically, when you look at these parents, I can see 3 capital B's, and capital B's means the dominant. So 3 capital B's out of the 4 means phenotypically, when I look at the parents, they're going to be 3 black-eyed parents is to 1 blue-eyed parent. That's my phenotype. I'm going to see it phenotypically. So my phenotype percentage is going to be 75% black-eyed children is to 1. It's going to be is to 1 or 25% blue-eyed children. That means, it doesn't mean that every family must only have 4 children. Even if the children are 16, it means 3 out of 4, that means 3/4 of the 16 children will be black-eyed. And 1/4 of the 16 children, 3/4 of the 16 children, and you'll work it out with a calculator, and that will tell you how many children will have black eyes. And then 1/4 of the 16 children, if there are 16, will have blue eyed, and you'll work it out how many of them will have blue eyes. So here is the genetic cross. P1 phenotype, use the words from your question. Next, represent your alphabets, your genotypes, right? Which is dominant, which is recessive. Is it homozygous dominant? 2 capitals. If it's homozygous recessive, 2 small letters. Is it heterozygous? 1 big, 1 small. Then meiosis, you're going to take 1 allele and put them down there. Put them as gametes, okay? After gametes, you put your Punnett square or your genetic cross, you write fertilization on the side, and then you describe the zygotes in the Punnett square. Genotypically, how are they genotypically means in terms of your dominant and recessive, heterozygous and homozygous. Then phenotypically, you're going to look at the dominant ones and say how many in the phenotype of physical appearance, how many will there be in percentage, and then that works out your ratios. Okay, so the next one is the visible trait is the phenotype. The genetic makeup you can see is your alphabets here, and then your alleles separate during meiosis to form your gametes. We'll see that very nicely, that during fertilization each zygote gets 1 allele from a parent. Now let's look at your sex-linked disorders. And your sex-linked disorders, learners, you must remember that on your karyotype chart, you know what's a karyotype chart? It's a chart that shows you the chromosome size and numbers, right, from pair 1 to pair 23. So chromosomes arranged in homologous pairs, chromosomes arranged in homologous pairs from pair 1 to 23 according to their size and their shape, okay? According to the size and their shape. So chromosomes arranged in homologous pairs according to their size and shape, and in a human karyotype, it's going to be from 1 pair to 23 pairs, and the 23rd pair is always going to be what? It's going to be your gonosomes, which is an XX in a female or an XY in a male. Now this is how you represent a male, XY, and a female with a circle and a plus, that's a female. So all females have in the 23rd pair. What's the 23rd pair called? Gonosomes. Females gonosomes is XX. Male gonosomes is XY. And that is your gonosomes on the 23rd pair. And from pair 1 to 22, what are they called? Autosomes. And how many autosomes will we have? We're going to have 22 pairs or 44 autosomes. Remember now in meiosis, we did pair 21. If there are 3 chromosomes instead of just 2, that extra X chromosome was due to what? Non-disjunction in anaphase 2, where the centromere failed to divide. And that extra X chromosome, you won't find it in the gonosome, you'll find it on the autosome on pair 21. That's where you're going to see that extra X chromosome, where the chromosome failed to divide and an extra one went into that gamete. So a gamete with 24 chromosomes will fertilize a gamete with 23 chromosomes, giving you a child with 47 chromosomes. And that disorder is called what? Down syndrome. Okay, so you see how you got to link meiosis to genetics. You cannot study it like a robot. You got to link and integrate your sections. Here now they are just showing you a father without hemophilia. What is hemophilia? It's a blood clotting disease. And it's a recessive disease. It's caused by a small letter. But because hemophilia is a sex-linked disease, it is found where? On the X chromosome of the 23rd pair. What's the 23rd pair called? The sex chromosomes, né? But we have a special name called gonosomes. So excuse me, learners. On the X chromosome, when there's a gene mutation, it's going to lead to a disorder called hemophilia. On the X chromosome, if there's a gene mutation, that gene mutation can cause a disease called hemophilia. There's many other sex-linked diseases, but in our syllabus, we're looking at hemophilia. And any sex-linked disease, we're going to put X, and because it's hemophilia, it's recessive, X to the power small h. they will give you the letter. They can put x to the power small h, they can put x to the power small g, they can put x to the power small t, if that sex-linked disease is caused by a recessive allele. How do we know it's sex-linked? We put x to the power, and whatever that letter is, they will give you. Okay, here they're saying this father is normal because he doesn't have hemophilia. Here they're saying the mother, she is carrying hemophilia. Now when you see a female, you don't know if she's carrying a blood clotting disease. You don't see her blood. She's not having her blood all over her body. So you don't know. She looks normal to you, but genetically, in her genotype, she's carrying the hemophilia allele. Okay, so females are XX. So if she is normal, that means let's say we're using x to the power small h to denote for hemophilia. That means x to the power capital H is going to be normal without hemophilia. So a father is normally XY. So the father is XY. If he is normal without hemophilia, he is going to be x to the power big H, which means he's carrying the normal dominant allele because he's normal, dominant. You see him as normal. He doesn't have the disease. There are no alleles or genes on the Y chromosome. That's the father. So this female, she is a mother. She's XX because she's a female. But she's normal and she's only carrying the hemophilia allele. So she's normal, that's the gray part. But if she's carrying the hemophilia allele, then she's going to be like that, because that is the hemophilia allele. But phenotypically, she's normal, okay? So you can see now what will their children be like. First of all, you're going to see that, please learners remember, every child is a zygote and receives one allele of a parent, okay? So now, let us look at this because you're going to come across a question like this. So while we are seeing this, we can do it immediately now to save time. Remember now, here's the son. A son must get an allele. For him to be a son, he must get the Y allele. The mother can never give a Y allele. She doesn't have Y. So this son can only be a son because it's the father's sperm with a Y that donates the Y allele to the son. So he must have an X. That means the X must come from the mother. So the mother has two X chromosomes. This son is colorblind, né? So it means that she has actually donated her X to the power small h to the son. That's why he's affected. But he got his Y chromosome from his father. But because he's affected, he must have got this H. He inherited X to the power small h from the mother. Okay, from the carrier. She was carrying that allele. So she gave that to them. Then here's a daughter. They can have a daughter. The daughter has to be XX because all females are XX on the 23rd pair. So it means the father now can only donate an X to the daughter. The father cannot donate an X to the male because an organism can only inherit one allele. If it's a boy or a male or a son, he can only get the Y from the father and only the X comes from the mother. So now here's the female, okay? She is what? She is normal, but she's carrying it, which means she's got the normal allele from the father, which is X to the capital H. She can't get the Y because she's the daughter. So she can only get that from the father, X to the power capital H. That means because she's colorblind, that means the mother must have donated that one to her, okay? That's another possibility. So she is normal, but she's a carrier just like her mother. Right, and then let's look at this one here now. So that was actually now, you can see Y and X, right? And then X, Y and X again there, right? So there's the male now. This male is unaffected. He's completely normal. So again, the father donated his Y allele. But because he's normal, it means the mother donated her normal allele to the son. Therefore, he's a male, He inherited the Y allele, but he inherited the normal allele from the mother. Okay, and here's the daughter now. And you can see with the daughter, she is very normal. She's not a carrier. So she got her X to the power big H from the father. And she got the other X to the power big H from the mother. You'll see it nicely in the Punnett square how we actually get these possibilities. Okay, so let's go to the next slide now. We're going to look at blood groups. You need to know about blood groups as well for tomorrow's paper. I'm sure there'll be a question on blood groups, and it's very, very easy. Children, to represent blood groups, phenotypically, you say a person has blood group A. You have blood group B. You have blood group AB. You have blood group O. So in a population, there are 4 blood groups. 4 blood groups. Blood group A, B, AB, and O. 4 blood groups. Because you're describing them, that's the phenotype. To represent blood groups, we must use I to the power something. If I am blood group A and I am homozygous blood group A, I'm going to be an A, blood group A, blood group B, blood group AB are all dominant blood groups. Blood group A, blood group B, and blood group AB are dominant blood groups. So you're going to use the word capital I, which stands for immunoglobulin. It's like a protein found in your blood. So just think of it as I as immunoglobulin. So on my I, I'm going to say I am blood group A. So it means to represent it genotypically, my genotype, I to the capital A, right? But remember, that means I am blood group A. I inherited an A blood group from my one parent. And because I'm homozygous blood group A, here's it like this. It means one came from the mother, one came from the father. That's why I'm homozygous blood group A. I can also be blood group A, but I can be heterozygous blood group A. It means my one parent was blood group A, but my other parent was blood group O. Now you see, children, this blood group O is a recessive blood group. You must always have a dominant and a recessive trait for a gene. In cases of blood group, 3 blood groups are dominant, A, B, AB. The 4th blood group is O, and that blood group O is a recessive blood group. So because it's recessive, it's recessive means it's a minority. We use a small letter I, 2 small letter I's to represent blood group O. We use 2 small letter I's because capital I means it's a dominant blood group. So one small I means it's a recessive blood group. But small I represents what? The O blood group. It represents the O allele, small I. So here it means that this person is blood group A, it's dominant, but the other parent was a blood group O. So one parent donated the O allele, which is represented as small I. Okay, so this person with blood group A is heterozygous blood group A, and this person here is homozygous blood group A. You can also have a person who's homozygous blood group B. It means both his parents are blood group B. He inherited the B allele from the one parent and the B allele from the other parent. That's why he's homozygous blood group B. You can also be blood group B, heterozygous, means you inherited one B allele from your one parent and your other parent had the O blood group. Therefore, the O is represented as small I. That is the I allele, which is actually the O allele, but represented as small I in a genetic cross. This is blood group AB, and blood group AB is what? 2 dominant alleles. That means one parent must have been blood group A, and the other parent must have been blood group B. The parents here can never ever be a blood group O, because you cannot see any O alleles over here. For a parent to be blood group O, he must have 2 small letter I's. But now this person, AB, none of his parents could have been. This person with AB, he can never have a parent with blood group O. Okay, they may have been carrying the blood group, maybe have been carrying the blood group I. That means they could have been a carrier like this one, but they cannot just be blood group O. Okay, so a person with blood group A can have a parent who is blood group A or blood group B. They can either be heterozygous blood group A, or they could be heterozygous blood group B, right? But they can never ever be a blood group O as a parent for this one. Now, what happens if a person has a blood group O? That means we said that only when something is dominant, it's in capital letters. Now, how can a recessive blood group be O? How can you say now it's dominant? This person is O, you can see it, and we said only dominant with the capitals can be seen in the phenotype. But there is an exception with blood groups, okay? For you to see blood group O, you must have two small letter I's. The recessive alleles have to be together. That means homozygous recessive. When you see the O allele in a heterozygous state, you'll see the dominant allele showing in the phenotype. When you see the blood group A is dominant, now here you don't see the recessive allele because the dominant A allele is showing in the phenotype. So for you to be blood group O, you must have two small letter O alleles, okay? That means this person has got one allele, one O allele from one parent and one O allele from the other parent. And how can we do that? You can see I can put that parent there is blood group A. He mates with the person who is blood group B. But for him to be blood group O, he had to get what? That O allele from one parent and that O allele from the other parent. That's why. Can you see? He can be blood group O and his parents can be blood group AB because his parents have to be heterozygous. They must carry the O allele for him to be a possible a blood group child. Okay. Remember, an individual can only have one blood group. You can either be A or B or AB or O. Right. There are always two alleles that control a blood group for that person. Right. There are three different alleles that control the blood group in a population. Two alleles control a blood group in a person. But three alleles control blood groups in a population. It's allele A, allele B, and allele O. Okay? And there are four different possible blood groups in a population. Right. Now let's look at a question. Mr. and Mrs. Fonella are concerned that their baby girl does not appear to resemble either of them. They suspect that the baby were given to them at the hospital was not theirs. So Mr. Fonella is blood type AB and Mrs. Fonella is blood type B. And the baby was given is blood type O. Can you see that that definitely this baby is not theirs? Because for the baby to be blood type O, it means it must have two small letter I's. One I has to come from the parent. Can you see? If Mr. Fonella is blood group AB, he is not carrying the I allele, so he could never have donated that to his child. So therefore, give the possible genotypes of Mrs. Fonella. She is blood type B. So the possible ones is she could be heterozygous blood group B because she's blood group B, or she could be homozygous blood group B. Okay? And let us see that. That is her blood groups. What will be the possible genotype of the baby girl? If she is O blood group, she can only be what? Two small I's. Let's see if it's correct. Yes. Explain why the baby girl with blood type O cannot be Mr. and Mrs. Fonella's daughter. They must have swapped her in the hospital when they were feeding her. She cannot be because the baby inherits one allele for blood type O. Right, from each parent. For the baby to be blood type O, you got to inherit one O allele, which is I allele from each parent. Since her genotype is two small I's, and Mr. Fonella doesn't have an O allele, therefore, because the father doesn't carry the O allele, that cannot be their child. Okay. Explain why the use of blood type for paternity testing is not conclusive. We can't just use blood groups to test the parent of a child, because many parents, many fathers could be blood group AB. Many fathers could be blood group B or A. So, because many people have the same blood group, we cannot rely solely on blood groups to determine paternity. So it says blood types can only be used to exclude the parent. Can you see what we did with Mr. Fonella? We said that child can't be theirs because he's blood group AB. He's not carrying the O allele. You see, therefore, that parent cannot be the possible parent for that child with blood group O. So we exclude him as a parent because he's not the parent of that child. Reason, he's not carrying the O allele. So blood type can be used to exclude a particular parent or a man. But it cannot confirm that the man is the father because many people have the same blood groups. Using your knowledge of sex chromosomes, explain why the sex of a child is determined by the male gamete. And you can see that normal females have 2 X chromosomes. So why is the sex of a child determined by the male gamete? See, a female is XX. So the father can donate the X and the mother can donate the X. But for you to find a child, a boy child, if you want a boy child, then the father can only donate, the father's sperm can only donate the egg, the Y. And the mother's ovum, in other words, the mother's ovum is going to be X, right? Okay, this child is a male. That means the mother has donated the X chromosome and the father can only donate the Y chromosome. So if a mother-in-law is squealing and saying, I want to kick my daughter-in-law out because she's only giving me daughters and I want a son to inherit the business, it's not the daughter-in-law that's a problem because she's only going to have X. And whether you're a boy or a girl, you're going to have an X chromosome. But who determines whether you're going to have a son? It's the sperm. Remember, sperm cells, 50% of the sperm cells are 22 autosomes with all X. And the third one, the 23rd one in a sperm, which is a gamete, it's going to have half the chromosome number. Now, out of 44, it's 22. But out of the 2 gonosomes from the sperms, 22 can have 22 plus X, and the other 50% of the sperms would be 22 plus Y. So it's only the sperm with the 22 plus Y that's going to fertilize an ovum, which is always 22 plus X. An ovum comes from the mother. A sperm comes from the father. So sperms can be 22 plus Y or sperms can be 22 plus X. But the ovum is always 22 plus X. So if there's going to be a daughter, it's going to be 22 plus 22 giving you 44 and X plus X giving you XX, which is the female. And if it's a sperm cell that's going to fuse with this ovum here, it's going to be 22 plus 22 is 44 and Y plus X is XY. So that's going to be a son. So it's the male gamete that determines the sex of the child, whether it can be a girl or a boy, because only sperm cells have either an X chromosome on the 23rd pair or a Y chromosome on the 23rd pair in a gamete. All right, so that's how you explain it. Now we go to pedigree diagrams, and these diagrams here, children, remember, you must look at the parallel side of it, the horizontal line. So that represents 1 generation, that represents, now here's another generation, 2, 3, and 4. They're on 4 different levels. See, that's the first level, second level, third level, fourth level. So this pedigree diagram is showing you the inheritance of genes over 4 generations. This means now, this circle is attached to the square. It means those 2 are people who mated, and this horizontal line or vertical line means these are the offsprings of that parents. And then that is the line that goes there. This horizontal line means that this person who's a male is marrying another person from another family. So you find that's the one, that's the parents, and these are their children. That there's another horizontal line means it's coming from another person now. This circle is a female. All circles are females. It can be granny, daughter-in-law, daughter, or mother. All squares are male. Could be father-in-law, grandfather, son, or father. Okay? Right. So this is a pedigree diagram. What does it show you? The inheritance of genes over generations. Okay. Now this one is particularly talking about non-hemophilic. Remember, if you're non-hemophilic, it just means you are normal. It also means you are unaffected. If you're a non-hemophilic male, it simply means you are an unaffected or a normal male. Right. So here, remember that is a square, it's a male, and a female is a circle. This is a shaded one. This is a key in a pedigree diagram. You must always get a key. Now, hemophilic, you know, is a sex-linked disorder. It's on the X chromosome and it's recessive. It's caused by a small letter. Right. Now, the shaded ones mean all these, this family tree, the shaded ones means they have hemophilia because all shaded are hemophilic. They're carrying the disorder, okay? So here you can see that all the females in this family, all the females here are unaffected. And it's only the males that are affected, okay? And what will that be? X, Y. Let's look at the male's genotype. It's going to be XY because it's a male. And we know that because it's hemophilia, it's caused by a recessive. I can put it like that. So all these males are going to have that genotype. All right. So for looking at a pedigree diagram, you look at the key, you look at who's normal, who's not normal, who's affected means it's carrying the disease. And if you are normal, it means you are unaffected. And then decide now, is the affected one caused by a dominant allele or is the affected person caused by a recessive allele? If it's a sex-linked disorder, then only you're going to use XX or XY. All right. Now let us look at this question. Gold syndrome is a sex-linked disorder. It's caused by a dominant allele. All right. Give Gabby's genotype. Now look at Gabby. Gabby is affected, okay? She's an affected female. And if she's affected, she's going to be what? XX because she's a female. And it's going to be small g, small g. Can you see that? Because Gold syndrome is a sex-linked disorder. Okay, let's look at it again. Gold syndrome is a sex-linked genetic disorder caused by a dominant allele. So now, if she's going to be affected, she's got Gold syndrome. You see, it's a sex-linked, it's another one, not hemophilia. But it's caused by a dominant allele. So she's going to be what? Big X, big capital X to the power big G, because it's dominant and it's causing the disease. So she's going to have on both of them big G. But now let's look at her children. This Pete and Gabby got married. Some of their children, the son and the daughter, are affected. That's shaded. Can you see? But some of them are normal. Remember, the mother is the one that donates her X chromosome to the son or the daughter. But the father only donates the Y to the son, but he can donate his X only to the daughter, remember? So she is going to be carrying the genetic disorder because it's the dominant XG. But because some of her children are normal, she can't be homozygous big X, X to the big G. She's going to be what? The mother, Gabby, is going to be X to the power big G. Yes, she's got the disease. But she's also going to be normal. She's going to carry the normal allele. All right. And there you can see that is Gabby's genotype. They have 4 children. Give the phenotype of their sons. Now here's their children here. Okay. So Pete and Gabby are parents. And then Pilusa is one of their sons. And Pilusa married Anju. And Pilusa and Anju, here they are. They have 4 children. They have 2 males and 2 sons and 2 daughters, okay? Give the phenotype of their sons. Now remember, Anju is the mother because it's a circle. All right. And then, because she is normal, what will she be? She's going to be XX. And because she's normal, she's going to be x to the power small g, x to the power small. She's not going to carry x to the power big G, because x to the power big G means you're affected, and she's not affected. She's normal. So she's going to be x to the power small g, x to the power small g. So now, what will the son be? The sons are going to be what? X, X. Sorry, the sons are not going to be X. It's going to be X, Y, X, Y. Ne? So what will the phenotype of the sons be? The sons are going to be now, because the mother is unaffected, the father is Pilusa, he's only going to be what? Donating his Y chromosome to the sons, isn't it? That means they're going to get what? They're going to get the X chromosomes from the mother. And the mother is only going to be like this. X to the, so it's going to be x to the power small g. So both the sons will be unaffected. Can you see that? They will be unaffected because they're getting the unaffected alleles from the mother. The mother has two small x. So whether it's that one or that one, that's going to be going to the sons because the Y is coming from the father. So both the sons are x to the power small g, and x to the power small g is recessive, and recessive in this case means normal or unaffected. Okay, and that's how you will explain it, that Pilusa is affected, right? It's x to the power big Gy, okay, here. And Anju is unaffected, she's normal, that's what's a chromosome. Genotype is males get the Y chromosome from the father, and therefore the x to the power small g is inherited from the Anju, which is the mother. Okay, so children, we are fighting for time, right? Here is the DNA profiling. Please learn about DNA profiling, where you're going to have the mother and the fathers, and here is the child's genotype. You're going to use a ruler and you're going to compare which bands of the child matches the parents. You can see that here the child, this DNA band matches the DNA band of father 2. Right, this DNA band, second band of child 2 matches again father 2. Then this, there's another DNA band of the child. This one matches the mother. Right, then that DNA band matches father 2, and this one matches the mother. All right, and this one in the child matches father 1 and father 2. Certain places of the DNA profiling, there are some common DNA bands in people, but there are some unique ones that are very specific to that family. Okay, here, although it's matching father 1 and father 2, it's fine, but it seems that only this one genetic band of the child matches father 1. And then this one here, there's none of it that's found in the mother and or father. Remember, every individual has a unique DNA. Here, this one here, that child's DNA is matching that father 1. Okay, that is matching the mother. That is matching father 2. And that one is matching the mother. So if you compare now, 1, 2, 3, 4, 5, 6, 7. In father 2, 1, 2, 3, 4, 5 out of the 7 DNA bands matches the child. But in father 1, only 2 of the DNA bands matches the child. So father 2 is a possible father of the child, okay? And then the mother, you can see she's got 1, 2, 3, 4, 5 genetic bands, but 4 out of her 5 genetic bands matches the child. So mother and father 2 could be the possible parents of the child, all right? Because now we're looking at a DNA profile. You can also do a blood test to confirm it as well if we know the child's genotype or blood group. Right, so now we already did this one here, learners, about, you can see this is how you represent that genetic cross by saying phenotype, okay, with the hemophilia. You can say you're describing it, then you put genotypes, you use the alleles, then you write meiosis, then you put the gametes, then you put them in a Punnett square. And you describe that, okay? You're going to have a worksheet that shows you all this, so you can go through it. In a dihybrid cross, remember there's 2 genes here, and you're going to have 2 separate alphabets. Big B and small b showing the hair color, and another alphabet showing the length of the hair. So the 2 genes are hair color and hair length, but the alleles are 4 alleles. Black allele, brown allele, short allele, long allele. And then you look at it here, children. For hair color, black is the dominant and brown is the recessive. For length of the hair, short is the dominant allele and long hair is the recessive allele. Okay, so this type of genetic cross is called the dihybrid cross. The genotype of the parent with black long hair is going to be what? Capital big B, and long hair, small h. Okay, and the genotype of all the possible gametes, that means of brown long hair, if you brown and long hair, it's going to only be small b, right? And therefore the gametes are going to be like that. Okay, and you can see all the gametes there are going to be one allele from each gene, small b, small h, small b, small h, small b, small h, like that. That's the possible gametes. And now, children, we're going to look at quickly the neurons, right? Please note the divisions of the nervous system. It's your central nervous system that has brain and spinal cord, which looks like a lollipop, and outside the brain comes cranial nerves, outside the spine comes spinal nerves, and because it's on the outside, that forms your peripheral nervous system. And this peripheral nervous system can actually go to form your autonomic nervous system, right, which is divided into your sympathetic and your parasympathetic nerves. Please learn the importance of the functions of your sympathetic. Always brings about a stimulating effect. It prepares you for an emergency, pupils dilating, heart beats faster, bladder relaxes, and your parasympathetic always slows down the responses. Here is the division of the nervous system. Nervous system, central nervous system, brain and spinal cord. Your nervous system is divided into the central nervous system and the peripheral nervous system. The peripheral nervous system means outside the brain is cranial nerves. Outside the spine is spinal nerves. Your cranial nerves and spinal nerves can also form your autonomic nervous system, which is divided into the sympathetic and your parasympathetic nervous system. All right. And you can see A is the peripheral nervous system, D is the autonomic nervous system, the type of nerve found at C is spinal, and then E is your parasympathetic nervous system. Please know the functions of the brain. A is the cerebrum. They're asking for the letter and the name. Give the letter and the name. You get 2 separate marks. A is the cerebrum, responsible for what? Interpreting your senses, right? It also is responsible for involuntary actions, not movements. Okay, that is your cerebrum, and responsible for higher order processes like thinking and intelligence and reasoning. B is your corpus callosum. It partly or partially joins the 2 hemispheres of the brain. It partially joins the 2 hemispheres of the brain. And then this one is the pituitary gland, which secretes your hormones. And then this here, just below the pons, the swollen part, is your medulla oblongata, which controls your breathing and your heartbeat. And learners, E is the cerebellum, which controls what? Your voluntary muscular movements and your balance as well, and your posture. Remember? Okay? And then you're going to go to all those parts. I've already discussed it, right? I've discussed all those parts, the heartbeat, the medulla oblongata. The parts of memory is your cerebrum. And we're going to look at the eye. Very beautiful picture here. This is the sclera, which extends to the cornea. Cornea is always convex, right? And you remember that behind here, behind your cornea is your iris. And then you have your lens, which is held by your suspensory ligaments, which is attached to your ciliary body. So near and far vision is called, the ability to see far and near object is called accommodation. Please learn it from your SIP material that for near vision, my paper is near, my ciliary CC, ciliary muscles contract, my suspensory ligaments will relax, my lens becomes more convex, and a clear image of the near object is formed on my retina. Then for far vision, distant vision, what happens? My ciliary muscle is still there, but now it will relax. Okay? My ciliary muscles will relax, and my lens becomes less convex, less refraction, more straight lines, and a clear image of a far object is formed on my retina. So this is very, very easily explained. And here you can see this is a pupil. It looks like a dot, but it's an opening. It's dilating, constricting, dilating, constricting. You can't control it. It's controlled by involuntary muscles of your radial muscles and your circular muscles. Remember, you're writing your paper in bright light, okay? So in bright light, CC, circular muscles contract, radial muscles relax, pupil constricts, and the opposite happens for dim light. That's the pupillary mechanism, okay? And you're going to look at the eye here. So there's some questions on the eye, and you can see that the diameter of the pupil is increasing at 6. Then between 20, can you see? Between 20 and 30 seconds, the pupil moved from dim light to bright light. Why? Because his pupil was quite wide. That means he was in dim light. So between 20 to 30 seconds, he took a walk into bright light. Then the pupil size constricted. So which structure in the human eye is responsible for the changes? Remember, the pupil is only an opening. What causes the pupil to constrict and dilate is the iris or the radial and the circular muscles. During which time was the person moving from dim light to bright light? He was moving from dim light to bright light between 20 to 30 seconds. Can you see that? Because it was. Describe the changes that took place in the eye that led to the diameter that indicated from 30 to 35 seconds. 30 seconds, his pupil was constricted, ne? Then he started to dilate. That means the person was now moving from bright light back into dim light. See, the radial muscles contracted, the changes that took place in the eye. He moved from bright light to dim light. And what was happening now? Now his radial muscles will contract, his circular muscles will relax, and his pupils will dilate. Okay, now the ear, this is hearing. This is simple, children. We talk about the pinna trapping the sound waves, passes it to the auditory canal, which causes the tympanic membrane to vibrate. The ossicles then are the malleus incus stapes. You must know it like that. They will amplify the sound vibrations, transfers it to the oval window membrane, and the oval window membrane creates pressure waves in the cochlear fluid, and that the organ of Corti is the receptor which converts your pressure wave stimulus into a nerve impulse carried by the auditory nerve to the cerebrum, where sound is interpreted. And then we talk about balancing. Remember, you have your semicircular canals, right? And they have a cristae, which is sensitive to the speed, to the changes in the speed and direction of the head. And that sends a nerve impulse to the cerebellum, which sends a nerve impulse to your skeletal muscles to maintain balance. You also have inside your inner ear a macula, right? The macula is the one that's sensitive to the position of your head. It will create a nerve impulse sent to the cerebellum, to your skeletal muscles to maintain your balance if you have to fall over, or if you're walking on a tightrope, or if you're somersaulting. How do you maintain your balance? The two receptors, cristae, which is sensitive to the position, changes in the position of the head to gravity, and the macula, which is sensitive to the position of the head. All right. So here you have your receptors, your cristae, your macula, and your organ of Corti, which is sensitive to your pressure waves. And this one, position of the head to gravity, and this cristae is responsible for sensitizing the speed and direction of head changes. Okay, and here's the questions here, children. This is part C, the auditory nerve. If it's damaged, it won't be able to carry a nerve impulse to the cerebrum. Therefore, hearing will be effective. Part A is a round window. If it becomes hard, it will not be able to vibrate, and therefore, it will not be able to absorb pressure waves, and sound can be affected. All right? So that is all the questions there, and you can actually look at it into your worksheet. Right. And then we're going to look at what is happening of middle ear infections. If you have a middle ear infection, that means in your eustachian tube where your ossicles are, that's going to increase your pressure in the inside of against your eardrum. So if you have a middle ear infection, mucus will build up. It will block the eustachian tubes. You won't be able to equalize pressure on either side of the eardrum, right? and it can cause your tympanic membrane to burst. And if it bursts or tears, then sound vibrations will not be able to be sent into the inner ear. It will lead to hearing loss. And learners, that brings us to an end. Please learn that this is a protractor. It's used to make a pie chart. You must have a calculator. You must add up all your values in your table, right? It must give you 100, and then you take each one, like let's say 15 out of 100, and multiply it by 360 degrees to actually get a sector or the value for a sector in a pie chart. You must have a sharp pencil. You must have a that's a protractor. That's a compass, which you put into your pencil in there, and you must have a ruler to actually measure with the radius of a pie chart. All right, you can also use a cup if you do not have a compass, because compasses are very dangerous. So children, use your mind the gap, use any material that you have to study the nervous system. Please learn the neurons, the sensory neuron, which carries the impulse from the receptor to the central nervous system. And then you have your interneuron, which is within the spinal cord. And remember, the gap between neurons is the synapse. It allows the impulses to move in one direction. Learn about Alzheimer's disease. It's a degeneration of the cerebrum, which causes memory loss. Learn that the myelin sheath of the neurons, if it is damaged or degenerated, leads to multiple sclerosis. Also learn that motor neuron carries the nerve impulse from your central nervous system, spinal cord, to your effector to bring about movement. And if should your motor neuron be damaged, you will not be able to move your leg or your limb, but you will be able to feel the pain. Okay, thank you, learners, and learn well and study well and do well for tomorrow's exam. Good luck. Thank you.